How do solitary mammals compare with social relatives on a morphological level? In this analysis of mammalian morphology, we compared sets of closely related animals from a variety of groups. From Felidae to Hominidae, we scoured the skulls of these organisms, searching for clues that could inform us on the sensory and social capabilities of these mammals. Does being social require a large brain? Does acute hearing require a large brain? We sought to answer these questions. Our first null hypothesis states that there is no correlation between the social ability of a species and its cranium size proportional to skull length. On the other hand, our alternative hypothesis states that social animals will have larger craniums in relation to skull length. We hypothesize that this is because social skills like keeping track of social relationships, communicating with others, and networking requires a lot of brain power. Thus, we believe that social animals would have larger brains and thus would have larger craniums to encase them. Our second null hypothesis states that there is no correlation between a species' sociality and the size of its sensory bone structure in relation to skull length. Being a social animal is quite the advantage because 5 individuals with less acute senses work a whole lot better than 1 individual with sharp senses. Having peers you can rely on to carry some of the load makes everything exponentially easier. That's why our second alternative hypothesis states that social species would, on average, have smaller sensory bone structures than the solitary ones. When it comes to solitary animals, acute hearing, smell, or vision can be the difference between life and death. However, when you're social, you can split up the work.
But before we could even start developing our project, we ran into a problem. When we were researching social animals and their relatives, the definition of the word social, or at least the way we were seeing it used, seemed to have really loose parameters. So, in order to keep our project as controlled as possible, we needed to lay out what we meant when we said social animal. Our definition of a social animal is an animal who tends to associate itself in a group and perform daily activities with that group. It is in this group which the animal builds a relationship that is more complex and nuanced than just with a mate. A relationship that extends beyond mating includes living in the same area, performing daily activities like hunting, gathering, or raising young, and consistently groups together with other members of the species.
When it came to our methods, we went virtual. First, we used a resource called TimeTree to identify relationships between social species and their solitary relatives.
Then, we searched through online databases such as Morphosource to find the 3D models of skulls of the species we found with TimeTree, like this chimpanzee skull. The great thing about Morphosource is that it allows us to find open access models of skulls that we didn't otherwise have any access to. We opened these 3D models in a software called MeshLab. In MeshLab, we plotted points on the skull's natural landmarks and measured the distances between them to quantify the skull's traits. Some landmarks we measured were eye socket length, width and height, distance between eyes, anterior to posterior cranium length, inferior to superior cranium height, lateral to lateral cranium width, lateral to lateral skull width, and much, much more. Trust me, it was much, much more. Equipped with our data, we went straight to the spreadsheets and organized, averaged, and visualized our newfound numerical information. Using bar graphs and scatter plots, we were able to find
numbers. Lots of numbers. Because we were dealing with raw data, it was almost impossible to figure out correlations at this point. Also, we encountered the challenge of comparing data across species. A whale skull is a little bit bigger than a porcupine's. To solve this problem, we had to use ratios, which would allow us to compare the proportions of these animals rather than the actual numerical numbers.
Even with the processed data, no trend between different groups was evident in regards to the first hypothesis. This could have been expected, given that these animals are separated by millions of years of evolution. Ultimately, we were left with no consistent trend between sociality and cranium size. In fact, we realized that what group the species belongs to matters much, much more than whether or not it is social. Thus, we are forced to accept our first null hypothesis that there is no correlation between sociality and cranium size. Similarly, we were forced to accept our second null hypothesis, due to the fact that there was no clear correlation between sociality and sensory bone structure measurements among groups of species. However, in the midst of a sea of evidence in favor of a null hypothesis, we derived an interesting reaffirmation of the well-known fact that primates are adapted to have their eyes close to one another. So even though we weren't able to find any new trend, it's clear that this research is in line with pre-existing scientific knowledge.
That being said, our project faced a lot of limitations. In fact, it was riddled with them. For starters, remember when we said that we went virtual for our project? Well, turns out that was a double-edged sword. Trying to navigate the 3D model of a hyper-detailed chimpanzee skull that makes even the most robust of computers struggle under the sheer polygon count is soul-crushing. Don't like that skull you got? Well, too bad. It's the only one in public domain and the museum's collections are just out of reach. Our sample size was quite small because of incomplete skulls, scarce 3D models, and technical difficulties. Another difficulty was the lack of resources available. Do you know how many in-depth labeled diagrams there are for the ear of whales? Not too many. Though, perhaps there is a beacon of hope in the lack of data and resources we had. With more time and a larger sample size, perhaps a clear trend would emerge. Over the course of millions of years of evolution, bones change a lot, making identifying the smaller, less studied areas of the skull a new difficult task for each pair. And when you're done identifying said tiny indent in the skull that does something, probably, you might finally come to the realization, hey, isn't this more of an organ thing?
Thank you to the American Museum of Natural History, Francis Forrest, Benny Heredia, Cici Messi Nieto, Cassandra Rodriguez, Brian Levine, and our fellow Lang students who have helped us through thick and thin. Also, a huge thanks to the 3D model of a chimpanzee skull that made our computers sound like jet engines.
If you have any questions, feel free to email us at the address above, ask in the breakout session, or send us a message by passenger pigeon. We hope you've enjoyed.